Okay, welcome back everyone to chapter 6. We want to pick up where we left off from the preceding lecture, which I believe was this slide that was pointing out some different cytopathic impacts that viruses had once they began to infect animal cells. Talk a little bit about viral infections and the fact that while we know viruses can potentially adversely affect host cells, i.e. causing them to lyse and rupture and die, there are other sorts of viral infections that can get into the host cell and sort of take over, but the cell does not die as a result. In fact, that virus can lie in a latent or dormant state for sometimes months or even years. And when there's some sort of a trigger, like stress often is described as a trigger of some viruses that cause them to kind of kick back into their active cycle, whereby the cell begins to make more virions and begins to exhibit the patient some of the symptoms we associate with that particular virus. So we refer to those as latent infections. And some good examples are listed here on this slide: the measles virus, herpes simplex viruses that cause cold sores and other types, of course, cause genital herpes, as well as what are referred to as the herpes zoster viruses causing chickenpox and shingles. And there's a photograph of what shingles looks like on the surface of the skin. These herpes viruses lie dormant, as it indicates here, or latent in nerve cells of all things. And as I said earlier, they can sit there for months or often years in the dormant state, only to kind of rear their ugly head, so to speak. And shingles is a classic example of that, whereby an individual may have had perhaps a more mild form of chickenpox as a child, and then when they're in their 30s or 40s, they suddenly come down with this very painful shingles due to the virus, again, sort of getting activated, if you will, from that latent dormant state. And again, what triggers that can vary, but I know stress sometimes is thought of as a trigger, if you will. So we would be dealing with lots of viral infections, probably, wouldn't we? Because we're always under stress, it seems. Luckily, that isn't always the case, though. We don't get reoccurring viral infections typically. Well, we've talked about some cytopathic effects in the previous lecture. This is another example of how some particular animal viruses can cause an even more serious issue with regard to the host cell and the organism. And here we're talking about how a virus, an infection by a virus, can cause what's called transformation to occur within the cell. In other words, cancer results from this viral infection. So it has nothing to do with being subjected to ultraviolet light, which we know increases the risk of skin cancer. We're not talking about inheriting a gene that might predispose you, say, to breast cancer. We're talking here, of course, about a virus that you unfortunately are exposed to, and down the road, you get cancer as a result. And as we know, having talked about cancer back in A&P, we're describing a situation whereby a cell is dividing uncontrollably. Those ordinary regulatory processes within the cell cycle, which are usually well orchestrated and regulated, those safeguards are completely ignored now. So instead of having, say, periodic red lights, if you will, that the cell might encounter within the cell cycle, now all the red lights become green lights, and the cell continues to divide, and those progeny daughter cells continue to divide, and so on and so forth. And so this uncontrolled cell division is what we often characterize as a property of cancer, right? In addition to metastasis and angiogenesis and that kind of thing. When we talk about a virus in you and I that has this capability of transforming a cell into becoming a cancer cell, we refer to those viruses as oncoviruses. the prefix onco refers to cancer. So an oncologist is a cancer specialist. An oncogene is a gene, when expressed, would trigger this uncontrolled cell division. And I've provided you a link here to the CDC that talks a little bit about the human papillomavirus, which, as it indicates here, there are 6 known types of cancers that can result from exposure to human papillomavirus. And the one that you might be somewhat familiar with is cervical cancer. If caught early, this can be treated, and the individual, the woman, will be fine. But the key is catching it quickly. And so you perhaps have heard about a 2-series shot that an adolescent can have. They recommend these be given when the child is between 11 and 12, just before sexual activity begins, both boys and girls, to really pretty much, I won't say eliminate the chance, but certainly significantly reduce the chance of acquiring any of these cancers. And of course, with respect to women, cervical cancer is a real big concern and worry. We've all heard of individuals, maybe even in our family, who've unfortunately gotten cervical cancer, maybe died from that. So it's certainly a very, very important cancer that can be significantly reduced in terms of the likelihood of getting that with regard to human papillomavirus causing cervical cancer. Now, there are other reasons why a woman can get cervical cancer besides exposure to this virus, but parents now have the choice to have their son or daughter vaccinated with respect to reducing the likelihood of this. So something to be thinking about if you don't have kids yet, or if they're still young. I'm sure your physician hopefully talks to you about that, your pediatrician. Epstein-Barr virus, perhaps you've heard of that too. It causes a particular type of lymphoma cancer. And the other thing that I think this bottom box really speaks to is the fact that there are probably lots of cancers out there that could have as a root cause some sort of viral exposure or infection. We just don't quite know yet to what extent exposure to a particular type of virus could perhaps predispose you to getting certain types of cancers. So a lot of really good research being done on that right now, and I have not read a whole lot about it. I think it would be very interesting to research. Okay, well, one of the complex viruses we talked about in the last lecture was this group called the bacteriophages, or phages for short. You might remember that these are viruses that target bacterial cells in order to complete their replication process. And so, as we'll talk about, E. coli has been a model that's been used for the last, at least, I'd say 70 years with regard to learning how these bacteriophages work. And so, unlike our previous discussions about how viruses, have to get into a host cell and then take over the host cell machinery and provide it with a new instruction booklet, right, to make more viral outer protein capsids as well as more viral nucleic acid, and then repackage that and all that good stuff. With respect to bacteriophages, what they do is they will actually inject their nucleic acid into the cytoplasm of the bacterial cell. So there's no bringing in of the virus like we've talked about earlier, and you'll see this in just a moment. The bacteriophage actually injects its nucleic acid, and that is, again, the instruction booklet that the bacterial cell will read to help make more bacteriophage protein coats and more bacteriophage nucleic acids. Ultimately, what typically happens is once the viral particles have been assembled in the cell will lyse or pop, and out will emerge, of course, thousands and thousands and thousands and thousands of bacteriophages, which can go on and infect other healthy E. coli cells or other bacterial cells. It's not just E. coli, and the cycle can repeat itself. And this last bullet that I put on here in purple and in a large boldface is to kind of stress the importance of how invasion sometimes, however, can end up not causing the cell to lyse, and we'll talk about that in just a second, but provide the bacterial cell with the means to be more virulent, meaning more disease causing. And that's often a result of having acquired genes from the virus. Remember, the virus here is injecting its nucleic acid. So what we have discovered is that phages can actually change the genetics of the host cell. And in this case, we're saying making it more virulent. So let's talk a little bit about the different steps or stages that phage will undergo as it replicates within a bacterial cell. And these 6 steps that you see here should look familiar to you. They're very similar to the steps we talked about in the previous lecture with respect to viral biology. But there's a couple of little differences here that we're going to, again, speak to. The first step, of course, is adsorption. And you know what that is. AD, not AB, adsorption. Here's again where the virus will use its particular outer chemical molecules, its unique glycoproteins typically, and dock with the receptor sites on the host cell, which again, remember, is a bacterial cell in this case. And after that has occurred, and you can kind of see that this electron micrograph, this very interesting colored electron micrograph, shows these little yellow or green guys. These are the bacteriophages on the surface of this bacillus-shaped cell. Here's a magnified view. We've talked a little bit about the shape of these. They almost look like little lunar landers. If you go back and study the Apollo missions that NASA sent out to the moon back in the 60s and 70s, that's what this sort of reminds me of. This left-hand photograph is actually a shot of many, many bacteriophages on the surface of this bacterial cell. Now you might say, well, it looks like it's cracking in half or is being lysed. I think this is simply some damage that has occurred to the cell just in the process of taking a photograph. I don't think you're looking at lysis here. You're seeing really the invasion, if you will, of a bunch of bacteriophage on the surface of a single cell, very similar to what we saw earlier in the scanning electron micrograph. Once that genome is injected into the host cell will respond again by making more bacteriophage outer protein coats and more bacteriophage nucleic acid. Those get assembled and mature within the host cell, and ultimately, at some point in time, the viruses will leave the cell once the cell lyses. Okay, so let's kind of take a look at this process. You can just kind of check out the 7 or so different steps involving multiplication of the phage. So if we start with number 1 here, of course, is the adsorption process. Here is the injection of the phage or bacteriophage viral DNA. In this particular example, we're going to talk about DNA. Keep in mind, again, that the host cell here, all bacteria have a single chromosome in the form of a loop or circle. So there's the bacterial DNA sort of in purple, red, and we have the viral DNA in blue. Once injected, as we've said, the cell is going to respond at some point in time and utilize this viral DNA. It will ultimately transcribe it into messenger RNA. And as you know from ANP1, that transcription process is followed by translation of the messenger RNA. And with the help then of ribosomal RNA and transfer RNA, amino acids are brought in and the polypeptide is made. The polypeptide, again, is going to be the proteins that form the parts of the new virions that we're going to make. In fact, here we are assembling one bacteriophage. So all these tails, sheaths, fibers, and so on, capsid, this is protein. In addition to making the protein via transcription and translation, the host cell here is also going to replicate the viral DNA. So it gets transcribed into messenger RNA and proteins are made, but there's also replication of the DNA. You got to have that, right, in order to repackage it into a bunch of bacteriophages. And here they are, maturing in the E. coli cell, and eventually a time will come when the cell will lyse and out come our bacteriophages that we said go on to infect other E. coli cells. Let's watch a little video on this.
The first step in the replication of virulent phage, such as...
Okay. Bring up the volume a little bit for you.
The first step in the replication of virulent phage, such as T4, is attachment. Protein fibers at the end of the phage tail attach to specific receptors on the bacterial cell wall. In the penetration stage, the enzyme lysozyme, located in the tip of the phage tail, degrades a small portion of the cell wall. The DNA in the head of the phage is injected into the cell, and the phage coat remains on the outside. In the transcription stage, the phage DNA is transcribed into messenger RNA, which is then translated into phage proteins. Next, the phage DNA is replicated to make copies for new phage particles. The phage DNA is also the template for additional messenger RNA synthesis. During this stage, many copies of phage DNA are made, and the various phage proteins are synthesized. During the assembly or maturation phase, the phage DNA becomes packaged in the phage head protein, and the various components of the phage structure come together to form the intact or mature phage. Finally, a phage-induced lysozyme is synthesized. This enzyme digests the host cell wall, resulting in lysis of the host cell and release of the phage.
Okay, just a great animation that sort of reviews what we just talked a little bit about. Another aspect of bacteriophage biology, however, is not the immediate lysis and assembly of protein, for example, and nucleic acid of the phage, ultimate lysis of the cell. But here we're talking about how once injected into the host cell, the bacterial DNA, in this particular example that we've talked about, becomes incorporated into the chromosome of the bacterium. So remember we said earlier we had this circular piece of DNA. This is the chromosome of the bacterial cell in red. Here is again the blue DNA injected by the phage. And so what's going on here is the viral DNA is getting incorporated into the genome, into the chromosome of the bacterial cell. And that viral DNA here in blue can sit dormant, if you will, within the chromosome of the cell. We refer to this phage DNA, in this case the blue colored part of the chromosome, we refer to this as a prophage DNA. And that bacterial cell can divide and multiply, and when it does that, it's going to replicate its chromosome. So the daughter cells that are going to be formed from this parent cell, I'll show you a video of that in just a minute, are all going to carry the prophage DNA in them. There will come a point in time where this lysogenic state, where the viral DNA is sitting sort of dormant or latent, will kick into the lytic phase that we just talked about a few moments ago. Okay, we talk about this process of induction that will trigger the lytic phase. From the lysogenic state, we get into the lytic state. And there's a number of different things that can cause induction. Again, triggering the lytic phase and the release ultimately of a bunch of viral particles. So let's watch a video on this.
When phage lambda infects E. coli, either the lytic or the lysogenic cycle may be followed. In both cases, the first step involves the phage attaching to the host cell and injecting its DNA into the host cell. In the lytic cycle, phage nucleic acid is replicated and phage genes are expressed, resulting in production of phage proteins. Mature phage particles assemble and the host cell lysis, releasing the phage particles. In the lysogenic cycle, the phage DNA is not replicated or transcribed. Instead, the phage DNA integrates into the host cell genome. The host cell can then replicate, carrying the integrated phage genome. The integrated DNA is referred to as prophage DNA, and the host cells carrying the prophage DNA are said to be in the lysogenic state. When the cells are exposed to ultraviolet light or to certain chemicals, phage induction occurs. The prophage DNA is excised and the phage enters the lytic cycle.
Okay. So this process of lysogeny, or the way in which, again, the viral DNA is incorporated into the into the chromosome rather of the bacterial cell. Remember, the host cell is not showing any ill effects, if you will, of lysogeny. It goes about its business, it replicates. The daughter cells that are formed from the parent cell, as we've said, are going to carry that prophage or prophage DNA. But there will come a point in time, as the video mentioned, the exposure to UV light or certain chemicals that can induce, again, the lytic phase. But what I want to talk about here is what I alluded to a little bit earlier with regard to how sometimes invasion by phage DNA into a host cell can make it more virulent. Remember me mentioning that about 4 slides ago? So what can sometimes happen is when a bacterium receives DNA from a virus, from a bacteriophage, it can acquire genes that allow it to become more pathogenic. Maybe those genes provide that cell with the ability to create some toxins that it wouldn't ordinarily know how to make, or particular types of chemicals, they mentioned here enzymes, that perhaps allow it to dock with and or invade a host cell, like your host cells. Sometimes bacteria need to be able to get into host cells to cause problems. So this process of lysogenic conversion is basically what occurs when a bacterium can become more pathogenic as a result of acquiring genes from phages. Pretty interesting stuff. So we can see again how a bacteriophage, which seemingly might not seem too worrisome to you or me, could become worrisome if it ends up inducing this lysogenic conversion of bacteria that maybe weren't pathogenic prior to being invaded by virus. And I give you some examples there of a couple different bacteria. Vibrio cholerae, that causes cholera, is one such bacterium that can undergo this lysogenic conversion. Clostridium botulinum causes botulism. Maybe heard of that before. So it's a constant concern that we have this process of lysogenic conversion. This particular slide has a photograph that you saw a little bit earlier, if you recall, and I made comment about how I thought perhaps this was not so much showing lysis as it was, damage to the cell once the photograph, was in the process of being made and so on. But I just noticed in your book there, they do define this as a weakened bacterial cell that has ruptured and begun to release virions, or in this case, baby bacteriophages. So it's really showing two things. It's showing the adsorption of the virus here on the surface, bunch of virions, but also it's showing the emergence of newly made virions. So we have just finished talking about this first column and the various steps involved in the replication of phage viruses. This is the animal virus category or column that we talked about in the preceding Zoom lecture. So you've heard of this before. I'd like you to spend some time and kind of compare and contrast the differences here. This could lend itself to a question on exam 2. Could bacteriophages assist us in fighting infection and disease? Well, this is a different way of thinking of things. We just talked about how bacteriophages could induce lysogenic conversion in bacterial cells that could in turn make us more sick as a result of the increased virulence. Here I'm talking about, obviously, could they benefit us? Could they help us? And I'm not going to spend time having you watch the video now during the lecture, but I do want you to click on that hyperlink and watch this video. It's very, very, very interesting. So here we are talking about how can we use these bacteriophages to help us, to our benefit. How can we manipulate these things in terms of genetics, as you'll see about when you read or watch the video. So check it out. So how do humans grow these viruses? And we're going to talk specifically about animal viruses because those are the ones that we have to be most concerned about, since we are animals and many of these can cause disease in us. And so one way to learn about the biology of these animal viruses is to, of course, be able to cultivate them and grow them, really. And you can't plate them typically on a petri plate like we can bacteria. The technique or the process behind viral cultivation, if you will, in the laboratory is a lot more difficult than it is growing bacteria in a petri plate or on a slant or in a broth. Much, much higher tech, requiring more sterile working conditions, if you will. And you can kind of get that sense from this photograph of this technician who's decked out in a hazmat suit, and he's actually handling this egg, looking for specific changes that might have occurred to the embryo. And you'll be learning about just how many eggs each year are sacrificed to help grow animal viruses so we can study them and see how they impact the cells of animals, in this case, embryologic cells of the chicken or goose or duck or whatever it might be. Again, we're assuming that the viral impacts to these eukaryotic cells of the bird can also potentially cause the same or similar sorts of morphologic disease-causing pathogenicities in you and me. So we're going to talk about 3 different methods that are currently being used to grow, cultivate, and identify different kinds of viruses. And this first one is a fairly more recent technique, referring to the ability to utilize cell cultures or tissue cultures in the laboratory. So as it mentions here, just in the last 20 years or so, have there been some flu vaccines that make use of growing virus in tissues, in cell cultures in the laboratory, and then harnessing those to again make the vaccines that we can inject into people to protect them from that particular viral infection. So what we're doing in this particular example is we are growing mammalian cells in vitro, so in the laboratory, and then we subject these cells to virus and we look to see what effect they have on the cells. And so here we've got a petri plate. Now I know I mentioned a few moments ago you can't grow them in petri plates. Well, I kind of lied. You can. You don't necessarily use the same ingredients here that you would in terms of a bacterial agar or a growth medium, but you use other special types of growth media. And here we're showing here in purple the single layer of mammalian cells of some sort that have been subjected to the herpes simplex virus. It's been inoculated and then incubated for a particular time frame and then pulled out. And then we're going to examine this plate under the microscope. And obviously you can see here in the blue shaded areas we've got normal looking mammalian cells, a single layer of them. I'm not sure exactly what particular cell type this is, but these look like nice healthy cells. Again, well-defined nucleus, you can see here in purple, nice monolayer. Where we have the white blotchy areas are indicating where the cells have been destroyed due to the viral infection and ultimately the lysis of the cells. So we sometimes refer to these areas as plaque areas, indicating infection has occurred. So this again is one way that we can grow and study viruses, basically by growing cell cultures and then inoculating those cell cultures with virus and looking for these cytopathic sort of impacts. We can also grow the viruses. We can cultivate them once we've identified them and use them to our advantage in the manufacture of certain types of vaccines. This second method is the one that has been around the longest, and it involves the utilization of eggs to initially inoculate with virus and then to study what effect the virus has on the embryonic tissues that develop within the egg. And this is where they have to maintain really, really sterile conditions, because when you inject the virus, you don't want to be introducing bacteria or fungi, other sources of cells that could potentially cause some impact to that embryo. We want to see what effect the virus has. So this again involves the utilization of millions and millions of eggs over the span of a year. And this is showing a technician again decked out in a hazmat suit. Looks like she's maybe working under a hood here, which is a system that has a vacuum that sucks all the air out of this enclosure so that there's no introduction of potential contaminants. It's like a clean room. And she's injecting the virus into the egg, and I won't go into the biology here of eggs, but nonetheless, it shows a developing chick of some sort, and they will inject that into either the amniotic cavity that contains the fluid that is bathing the embryo, or they may sometimes end up injecting right into the developing embryo itself, or it may involve manipulation of this membrane that sort of surrounds the entire developing embryo, including, this is the yolk sac here. They even, as it mentions here, inject sometimes vaccine or viruses into that. And then, don't ask me how, but in some way they can obtain vaccines as a result of utilization of chicken eggs. And it obviously takes millions and millions, hundreds of millions of chicken embryos to make flu vaccine. At least this is the method that had been used up until about 10 or so years ago. But it was very labor intensive. It required lots of eggs, usually chicken or duck eggs they would use oftentimes. Turkey eggs are the most common choices for inoculation. So they would candle the eggs, they would look for embryonic deformations. They would often dissect the egg, look for what effect does it have directly on the embryo, and so on and so forth. You can check out this link. This goes specifically as to how vaccines like the flu vaccine are made with the use of eggs. More recently is this third method of cultivating, growing, and utilizing viruses, in this case by manipulating the genetics. So when we say recombinant technology, we're talking about recombinant nucleic acid technology. Nucleic acid, again, are DNA and RNA. You remember that some viruses are DNA viruses, some viruses are RNA viruses. And what this, in a nutshell, involves is not the direct introduction of the virus into you, or be that an attenuated, weakened form of the virus, or it could sometimes even be living virus. But rather here we're talking about the introduction of often nucleic acid into the host, into you, as is the case for the current Pfizer and Moderna COVID-19 vaccines. Now, I'm not sure about the AstraZeneca and the Johnson & Johnson vaccines. I'm not sure if they're RNA vaccines or not. I should know, but I just don't know. Something tells me they're not. So I can't really speak to those, but I can tell you, having just received my second Moderna shot 2 weeks ago today, in fact, both Moderna and Pfizer have come out with mRNA vaccines. Now, keep in mind, it typically takes 10 to 12 years from the time research begins on a vaccine to the time it's rolled out in use, widespread in the world. We've talked about, of course, this idea that in less than a year, we go from identification of the COVID-19 viral genome, which was known almost within a month or two of when the infections first began in China, back in November, December of 19, to here we are cranking out vaccine. These mRNA vaccines have been rolled out now for the last couple months. So when you get a Moderna or COVID vaccination, and you all know you need 2 shots, typically 2 weeks apart, although it can be a little bit longer than that. What you're getting injected with is a recombinant vaccine, like we're talking about here. And the mRNA that you're injected with will tell your cells to translate that instruction booklet, or that strand, really is what it is, that strand of messenger RNA. And what your cells will make are the glycoprotein spike that we know sits on the outside of the COVID-19 virus. Look at any diagram of a COVID-19 virus, you'll see spikes sticking out, right? We talked about it actually at the beginning of the previous lecture. So your body is producing the spikes because it has the instruction booklet here that it got in the form of messenger RNA from the vaccine. When you make the spikes, your immune system is triggered, just as if you had acquired the actual virus, in which case the normal spikes would dock with certain proteins, blah, 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 blah. We've talked about that, and this virus is allowed in. Only here you're not getting the virus. You're getting exposed to what the spikes look like, and your immune system is responding by producing antibodies against those spikes. Why would you want to do that? Well, because if you have the ability to produce antibodies against the spikes, if you're actually exposed to actual COVID-19 by inhaling it in the air because somebody near you had it and coughed or what have you, you would have this real robust immune response to the point at which the virus would have no chance of getting in and causing, an infection. That's the basic mechanism behind what that mRNA virus of either Pfizer or Moderna, how it works. So it's really interesting stuff. So in addition to using lots of turkey and duck and chicken eggs, there has been a lot of research also done on other mammals to see how they respond in terms of viral load. How do viruses grow? What effect do they have on host tissues? And this is not without its controversy, because nobody likes to see a cute little guinea pig or a rabbit or primate of some sort in a laboratory suffering the ill effects of having been injected with a particular virus that we want to learn more about, because we're concerned about how it's going to impact us. So we use animals because they're much closer in biology than, say, even an embryo, a bird embryo might be. And so for many, many, many decades, there's been a lot of research done on this. And I'm going to leave it at that. I'm not saying it's not a good thing. We have certainly benefited over the years thanks to animal research, whether it's open heart surgery or viral impacts and learning how it can influence mammalian cells. We certainly know that all of us have come down with viral infections in our lives. We've all had the flu at some point in time, for example. And so every year, lots and lots and lots and lots of people throughout the world suffer the ill effects of viral infection. Some people die as a result of viral infection. We have seen 500,000 plus people die in the US from the COVID virus. Ebola is making a comeback in recent years in West Africa. It was in the news about 8 or 10 years ago, but we were able to tamp it down and kind of isolate the virus, but now it's coming back a little bit. Hopefully that can be isolated, and we won't have near the number of deaths as we did 6, 7, 8 years ago. The fact, as I said earlier, that some cancers that we get could have viral underlying exposure issues. In other words, because you're exposed to a particular virus, you may have a predisposition to certain kinds of cancers. But look at these chronic diseases that we associate with perhaps more lifestyle decisions like obesity, overeating, or not having enough exercise. Well, perhaps there's some research to suggest that in some cases, obesity may be due to exposure to different kinds of viruses that triggered that, or type 1 diabetes, or MS, as it mentions here. So again, a lot of work still needs to be done in seeing if there is a direct correlation between viruses and certain long-term chronic afflictions. Very interesting to study. We use antibiotics to treat bacterial infections. And we know we do have some antibiotic resistant issues, antibiotic resistance issues with respect to that. And we'll talk more about that in an upcoming chapter. But as it mentions here, antiviral drugs are not nearly as abundant as antibacterial drugs. We have some antiviral drugs that we'll talk about later. But it's tough. It's tougher to control viral infections sometimes. You might wonder, why is that the case? Well, because many of these antivirals that we might prescribe are directly targeting different stages of how the virus influences you and me, our host cells, ranging from the adsorption step to the replication of the nucleic acid of the virus in the host cell, to the synthesis of more viral proteins, to the formation of the envelope on the inner surface of the cell membrane prior to budding. So you got to go back and you have to understand the steps involved in viral replication. If you understand those, then you can understand much better how certain antivirals are actually potentially affecting the function of the host cell in a negative kind of way, because they're trying to prevent the host cell from making more virus or allowing the virus even into the host cell. So those serious side effects need to be thought about. You cannot use the same sorts of drugs on viruses as you do bacteria. Antibacterial drugs, for the most part, don't influence our cells because we don't have, for example, peptidoglycan. We don't have cell walls. Some antibiotics like penicillin target the peptidoglycan formation. It doesn't allow it actually in certain kinds of bacteria. So that's not going to cause any problems to you and me. But when we're talking about drugs that influence host cell biology in an effort to control viral replication, then that's a side effect that we need to be thinking about. Okay, we're going to shift gears as we end chapter 6 and talk a little bit about prions and viroids. Prions are these very fascinating misformed proteins. Up until about in the 1990s, we did not have a real good sense of what these things actually were. We were kind of like Pasteur back in the 1800s. You know, he knew about these things that were not as big as bacteria. Remember, he worked on the rabies virus. That's kind of where we were in the 1990s. We knew we had this entity out there, and it wasn't viral, and it wasn't bacterial, but it was causing, you know, death and disease in animals. And so a lot of research was pumped into the study of these malformed proteins called prions. And what we discovered was many of these prions are extremely resistant to normal sterilization techniques. So remember, we talked not too long ago about how the autoclave can kill not only the vegetative cells, but also the endospores, which are those, you know, desiccated resistant forms of certain bacilli. These prions, as it mentions there in bullet number 3, can stay viable at temperatures above 1100 degrees Fahrenheit. Now, that is extremely hot, yet these prions can survive that. So we have to be cognizant of that if we sometimes want to develop techniques to get rid of these. I can't say kill them, because this is a protein. Proteins are not alive. Viruses are not alive. Okay? Viruses are acellular. We mentioned that in the previous lecture. But this is a protein. This is an organic molecule. So it's not alive. So when we talk about getting rid of it, we would have to talk about denaturing the protein. What can we do to denature or disrupt the chemical structure, if you will, of the protein? Because remember, the structure influences the function, right? And so I'm going to talk about a group of prion diseases that are referred to as spongiform encephalopathies. And these are all impacting the brain tissue and central nervous system of animals. Okay, it's typically the brain, but I would not be surprised if the spinal cord is not also impacted by this. So let's talk about a couple of these. Here's a list of several that we find in animals, and as far as I know, these prion diseases are pretty much exclusively found in mammals. I have not read about any prion diseases that influence fish or amphibians or other kinds of animals. These are mostly mammals, I guess. But all of these spongiform encephalopathies, what they do is they get into the healthy neuron, okay, which you know is a nerve cell. And so we'll depict these prions as these pink structures. They get into the neuron. When they come into contact with what we would call normal proteins, those that of course have the proper shape and they're providing a beneficial function to the cell, when they are exposed, when the prion proteins are exposed to the normal functional proteins, it causes them to become prions. They convert from a normal morphology, shape, function into an abnormal shape, abnormal function. So we sometimes talk about use the word co-ops. Co-ops the normal biology, if you will, or function of these proteins into prion, abnormal shaped and abnormal functioning proteins. What do I mean by abnormal functioning proteins? Well, here's what these prions can do. In large numbers, they can bind to neuronal cells, and they form fibers initially within the neuron and end up destroying the neuron itself. So in this particular stained slide, we can see the prion proteins here in sort of orange. This indicates where in the cytoplasm the prion fibers have been deposited. And what that does is it prevents the neuron from transmitting the impulse like it ordinarily would. You all remember from A&P2 the process of depolarization, repolarization of the neuron membrane and all that good sort of stuff. So instead of being able to transmit the impulse the way it should, these nerve cells not only are unable to to transmit impulses, but the tissues, the cells, the neurons die, and you get these big gaps in the brain, and then probably the spinal cord. Again, typically we talk about the brain. So here's what it would look like histologically. So here's normal cerebral cortex tissue. We can see the cell bodies of some neurons here, which should look familiar to you. These rounder structures are neuroglial cells, which are kind of supporting nerve cells. They support the multipolar neurons. And when these healthy, normal brain tissues are exposed to prion proteins, look what happens to them. You get these big gaps, which indicate the death of the neurons. First, they lose their function, then they die, and you get these sort of circular, microscopic circular gaps. This is where the term spongiform comes into existence. It's like sponge candy, big gaps. Instead of having nice normal neuronal tissue, you've got nothing there. And you can, of course, imagine what that does in terms of neural circuitry and such. You can't get normal sensory or motor function take place because there's nowhere to send the impulse to. There's no tissue. So these big gaps occur. In sheep, one of the first external symptoms of prion disease in a sheep is where the sheep begins to rub itself on a fence post or on a tree because it itches. It's very irritating surface of the body, and you can see how the fleece is beginning to wear off, and so you get this disease called scrapie. So the animal scrapes itself against the tree or against the fence post. Again, due to early stages of spongiform encephalopathy. So this is impacting not just the brain, obviously, as it will later, causing the sheep to die, but there appears to be some supplemental ancillary tissue damage or impact, if you will, with regard to the skin. Mad cow disease, you may have heard of that. There's a nice little YouTube video I'd like you to watch that describes more of the biology of these prions. So please watch that. So this bovine spongiform encephalopathy, bovine referring to cow, occurs when the cow, you might say, how does the cow get the prion? Well, believe it or not, most cows are fed in big yards or in local, farms. The feed that they sometimes give cows that you buy from a feed supply company, at least years ago, contained oats and hay and straw and all those good sorts of things. But there would also be fillers they would use. And in those fillers was ground up cows. And you're saying, what? When a cow is butchered, yeah, we save the meat and we make hamburger and steak and other roasts, other things out of it. But do you ever think about what happens to the spinal cord, what happens to the brain tissue, what happens to the bones and the cartilage and other connective tissues? Well, they don't throw that out. They grind all that together. And that becomes part of the filler that's used in addition to the hay and the straw and the corn and the wheat and all that stuff going into the cow feed. And yeah, as hard as it is to believe that, a lot of cows back in the mid 90s were fed not even contaminated food. It wasn't considered contaminated, but it had prion proteins in it from cows that had been butchered, and they had the encephalopathy. Whether they died of it or not, we don't know. But it got into the livestock feed. This cow eats the feed. It acquires the prion that way. It ends up with spongiform encephalopathy. And so when you compare a healthy brain here, and you can see this frontal section with a control label on it. So this is the healthy, non-infectious brain. Here's the brain of the cow that died of BSE. And what immediately comes to mind is the fact that the overall size of the brain is significantly diminished, and it's a good 15, 20% smaller in size. And the other thing that stands out, at least to me visually, is the enlarged ventricles. Remember the lateral ventricles you guys learned about in A&P too, that circulate the cerebrospinal fluid from the choroid plexi. So there's a marked difference, even a gross visual marked difference when you compare these frontal sections. If you were to examine histologic sections of brain tissue between these two, again, you'd see lots and lots of gaps, like we saw in the previous slide or so. And ultimately, the animal goes into a coma, their CNS completely collapses, and the animal dies. But in the process can exhibit some really strange behavior, and that's where the term mad cow disease comes into play. Cows are generally pretty docile animals, but their brains are so impacted by the prions that they behave very erratically, can get somewhat aggressive at times, and it's just very, very sad to see. They lose all coordinated skeletal muscle action. They don't walk very well. They're struggling to stay on their feet sometimes. It's really a sad thing to see. Well, this can occur in humans as well in a disease referred to as Creutzfeldt-Jakob disease. So this J is actually pronounced like a Y, Creutzfeldt-Jakob disease. And here we can see again the spongiform encephalopathy in a human brain tissue section. We can see the big gaps illustrating again where the prion has co-opted the normal brain protein, and the end result is the destruction of the neurons. And so many of those same symptoms that the cow would exhibit, that is sort of strange behavior, ultimate CNS collapse, central nervous system collapse, and the person can die. There's also a marked reduction in the overall size of the brain, like we saw in the cow. This occurs in people as well. And I'd like you to watch this link. This is a Mayo Clinic link. There's a video that's about 3 minutes long. It talks about a woman who came down with a genetic variant of Creutzfeldt-Jakob disease, meaning it's not that she ate contaminated beef that had the prion in it, and it's not that she was ingesting, the prion in any sort of way. This particular video talks about a fairly rare form of Creutzfeldt-Jakob that occurred as a result of her inheriting the gene from her mother or her father. The end result is the same. The prion, the malformed protein, the spongiform encephalopathy formed, all that is the same. Only she got it as a result of inheriting a particular gene that triggered the formation of the malformed protein. There were historical incidences of people in the 90s who did eat contaminated beef that had the prion in it, and they acquired the prion that way. But once that was diagnosed and determined as to what caused these people to suddenly have these bizarre, CNS diseases, they were able to trace it back to contaminated beef that were eating basically brain tissue of other cows. It's a fascinating story, kind of creepy, kind of weird, but this is what happens in today's modern farming. There have not been a lot of cases of mad cow disease anymore. I think we've done a good job in isolating it and controlling it, but interesting to study. The last thing I'll leave you with is another particular strange infectious agent that, similar to prions, is non-cellular. We're talking here about something called a viroid. And what they've discovered is that viroids tend to target certain kinds of plants. And you can see how the viroid causes some decolorization of these avocados, or definitely a reduction in the overall size of the potato tuber. as a result of exposure to a small little piece of RNA. Remember, this is a type of nucleic acid. As to how the plant acquires the viroid itself, I cannot speak to that, but simply you should know that these are, again, infectious RNA entities, little pieces of RNA that are found only in plants and do play some role in reducing harvest of particular plants. Okay, that wraps up chapter 6 on viruses.